All right, welcome. My name's Holden Maecker, and I'll be your fearless leader for today. I direct the Human Immune Monitoring Center here at Stanford, which is a core lab that I'm going to introduce you to a little bit in the first few slides, just because I think it's kind of an interesting concept that maybe some of you would like to even take advantage of or take back to your institutions. And then we're going to have a series of lectures this morning. I will give the first one on the fluid-ion biomark technology for nanofluidic qPCR. And then we'll have a couple other speakers doing technical talks on particular technologies that you haven't heard about yet in this course, because I know you've heard about CyTOF and PhosphoFlow quite extensively. And so this is the day where we kind of broaden it out and introduce some other technologies. After the coffee break, we're going to have a couple of talks that are more bioinformatics related, and that again, hopefully will give you some concepts that maybe don't relate directly to PhosphoFlow and CyTOF, but generally to the handling of large data sets and kind of systems analysis. So that's the morning. After lunch, we're going to have a series of demonstrations. They're in small groups. We're going to break you into 3 groups so that you can rotate between the first 3 demos. Those of you who want to stay on for the last one will head over to, or head down, I should say, to the basement of CCSR for the last demo, which will be on the nanofluidic immunoassay platform. So that's the overview. Let me tell you then, are there any logistics questions for today? All right, let me tell you then a little bit about the HIMC. We were started about 5 years ago by Mark Davis, who had the idea that it would be great to have a kind of one-stop shop for immune monitoring technologies. To help out clinical investigators, translational studies, with everything from designing the assays and the sampling points in a clinical trial, for example, to the processing and banking of those samples, the actual running of the immune monitoring assays across multiple platforms, so you don't have to go to several different core labs to get flow cytometry and genomics and immunoassays. And then the data analysis and interpretation, helping the investigators to put it all together. So kind of start to finish, the ideas that we can be a go-to place for that whole process. And in addition to this, which is sort of our major goal, we also have as a goal to kind of be a proving ground for new technologies. So because we have this constant stream of clinical samples coming through, and we're doing a lot of standardized assays on various platforms, it's a very nice place in which to compare new technologies, to bring them in, do them side by side, and get some relevant data on how they would perform in the context of immune monitoring. And then thirdly, we hope to actually do more than kind of the average core lab, which returns the data to the investigator and then never does anything more with it. We actually want to amass the data from multiple studies and be able to mine that to eventually create some new metrics of what immunological health looks like on these various assay platforms and how different kinds of diseases are distinguished based on those platforms and those metrics. So that's a kind of ambitious third goal that we have. Now, in order to kind of achieve this one-stop shop concept, we've tried to build some relatively comprehensive assays that we do on a regular basis that we consider our standard assays. And we kind of have to do this because if we built a custom assay for each new project that came along, we would never get anywhere. We'd spend all our time in assay development. And so being comprehensive allows us to meet the needs of many different projects with regard to the analytes that we're looking at. And it also serves that third goal of allowing us to then amass the data, because it's the same assay It's a relatively comprehensive data set, and so we can really gather data beyond that might have been intended for the original investigators' questions. And so this is just an example of some of the subsets or the analytes that we can look at by the different platforms. And actually now with the CyTOF replacing flow cytometry to some extent, we have many, many more subsets that we look at phenotypically and functionally. And part of the also of the goal here is to do assays that have a functional component that stimulate the cells in vitro and read out something beyond the resting state, because that's, as the Nolan lab has shown in a number of different disease states, can uncover things that you don't see otherwise. And so examples of those kinds of functional assays are listed here. And on that note, a little more detail about what we consider our standardized assays, again breaking them up into sort of three general areas of looking at cells, looking at proteins, and looking at genes. I've kind of listed here what some of these assays are, what their targets are, and I'm not going to go through them in any detail this morning, but you will hear much more about the cytokine assays, both fluorescence and electrochemiluminescence, in the next talk from Yael. And you've already, of course, heard a lot about phosphoflow and flow cytometry and CyTOF in general. What I'm going to talk about this morning is the Fluidigm platform for microfluidic qPCR, and so that's a kind of a nice segue into the rest of the talk. Before I do that, any questions about HIMC, anything that I can answer that you might be curious about?
So do you have a repository of kind of all the different protocols that you have for your standardized protocols?
Sure. Yeah, actually, on our website, if you go to there's like individual technology pages for immunoassay, for flow cytometry, and on those pages are linked protocols. So yeah, we do have that. And I should also mention that the database that we've used to try to accomplish this goal of amassing all of the data and mining it across studies is something that is going to be the subject of one of the demos this afternoon. So I'm going to do that demo this afternoon. Yes?
Do you do any tissue banking services, or is it?
It's by tissue you mean things other than blood cells, or just banking in general?
Yeah, other than blood cells.
Yeah, we haven't really. The Cancer Center is the main one that's been doing tissue banking here. But partly it's because we haven't really been asked. I mean, pretty much everybody comes to us looking for PBMCs and serum process, banking and such. Okay, so if there's no other questions on that, then we'll go ahead and start talking about qPCR. A lot of you may already know these concepts, and so I'm going to go sort of quickly through an introduction that, if it's already familiar, I apologize. But just so that everybody understands the basics, qPCR, or quantitative PCR, is a method of amplification of a gene sequence which is quantitative in the sense that it has a fairly linear relationship it gives you a readout that has a relatively linear relationship to the amount of the target sequence, as opposed to conventional PCR, which amplifies exponentially but doesn't tell you very quantitatively how much message you originally had. So in the version that we use, the readout is fluorescence, and you measure the accumulation of the target sequence by the accumulation of fluorescence, and that works basically by displacement of this probe, which has a reporter and a quencher molecule attached to it. This is the most commonly marketed system for this is called TACMAN. And the way this works is that the reporter is fluorescent only when it is cleaved away from the quencher. And so as the target sequence, which the probe is specific for, gets displaced, as the probe gets displaced from the target sequence, then the reporter, the link between the reporter and the quencher is degraded, and you get fluorescence, and that is a one-for-one kind of readout. You get one fluorescent molecule for each strand that is amplified. Okay, so that's the way the system kind of works. And then what happens, of course, is that you have to get to a certain threshold, a certain number of fluorescent molecules that are released in order to be able to see it in a particular well. And then once you get there, you start amplifying that fluorescent signal. And so if you read out the fluorescence as a function of the number of cycles of PCR amplification, of annealing and extension and dissolution again, then you get something that looks like this. You get no fluorescence detected until it reaches some threshold where you start to see the signal, and then it starts to go up, and it's relatively linear, although at some point it eventually saturates. And so the way that we try to quantitate this is by establishing what we call a critical threshold value. And I'll get back to this point in just a minute, but basically we try to establish a value which is a bit above the baseline or the noise that we have in the system, but tells us essentially when that particular reaction is coming up above the noise and into the readable zone. And the way that we quantitate it then is just by cycle number. So the CT or critical threshold value is the cycle number at which you cross this line. Here 17, 19, or 20 point something. It can be a fractional value. All right, but the CT value then, the bigger the CT, the less abundant that sequence was, because it took more cycles to get to the above the threshold. And the lower the CT, the more abundant the message. All right, so why do we want to do this kind of assay as opposed to, say, a microarray? It's certainly more quantitative than a microarray, which is simply an annealing. A microarray can get saturated relatively quickly and doesn't really give you a linear readout, at least not as linear or quantitative as a qPCR. And so a lot of people use it as a sort of confirmatory assay after they detect some difference between, let's say, a patient and control group on a microarray. They have a handful of genes for which there's a significant difference that they see, and they use the qPCR to now in a targeted fashion, confirm those results. And obviously it is more targeted. You're not going to do qPCR across the entire genome the way you would a microarray, but that makes it cheaper on a per sample basis. Not per gene, but per sample. And another advantage, which is more specific to the system that I'm going to talk about, where we're doing microfluidics, is that we require very little input RNA. I mean, this is true of qPCR in general. When you compare it to a microarray, you usually need less. The tiniest contaminant will cause an amplification in PCR, but it's especially true when we get to microfluidics, because we can actually go down to a single cell's worth of RNA. Okay, so why single cell qPCR? Which, as you'll see, is one of the kind of fortes of this microfluidic system, is the ability to do qPCR on single cells. Well, the main point is that you will often have systems in which you want to look at the heterogeneity of a cell population. And obviously, you've been learning about CyTOF as one approach to look at cell heterogeneity when you have single cells in suspension. But CyTOF is, of course, dependent on having antibody probes to the particular proteins of interest. It's easier to do for surface proteins than intracellular, but in any event, you need to have good antibodies. And the nice thing about qPCR is that it works for pretty much any gene. Now, it's not at the protein level, it's at the message level. So there is the potential for disconnect depending on translational regulation. But it is essentially an easy way to assess any particular gene's expression at the mRNA level. And so an example of the kind of heterogeneity question that you might ask is you're looking at T cells, and I happen to be a T cell immunologist, so I come up with T cell examples a lot, and I will take you through a more detailed one in a minute. But you might ask whether all of the T cells that bind a particular tetramer, so have a particular MHC peptide specificity, do they all have a similar phenotype? Okay, and so you could do single cell sorting. You could assess an array of genes by this method that I'm going to show you and ask about the gene expression profiles of each cell. It also is a great way to examine rare cell populations. So one example that actually came through our HIMC core a little while ago is a project that we did for Kari Nadeau's group looking at sorted basophils. So they have allergic versus non-allergic donors, and basophils are rare cells, and it was difficult to get enough of them to get enough RNA to do, for example, microarray. But with this technology, we could actually sort single basophils and not only get expression profiles on them for at least a limited number of genes, but we could ask about the heterogeneity of those basophils cell to cell. So even if they only could get 40 basophils from a given sample, we could analyze that and get essentially 40 replicates of data on gene expression. Okay. And then finally, to sort of finish off the intro, suddenly there's no response. Let's try that. Okay. Why go to the microfluidic level? And I've kind of already alluded to some of this, but let me give you sort of the three basic advantages that you have with microfluidic qPCR. One is that you use a lot less reagent. Okay? So because everything is in microwells, you don't need very much input RNA, and the primers, the buffers, everything is used in much smaller amounts than they would be for a conventional scale qPCR. And very importantly, you do a lot less pipetting. As I'll show you in a moment, you don't have to make every single combination of primer set reactions and samples. The instrument actually does that for you. And so you save a lot of pipetting time, and there's much, much less chance for an error in terms of making all of those combinations if you had to do that by hand. It's the kind of thing that you would never ask a technician to do because either it would A, drive them crazy, or they would make an error, or both. So that's going to be actually relatively important when you get to larger scale projects. And the final one, which I did already mention, is that now by scaling everything down to a micro scale, we make the analysis of a single cell's RNA possible. All right. Now I'm going to have to click it here. So the way this system looks is we have a chip which looks very much like a microplate, but it's actually a microfluidics chip. It has input wells that are compatible with a multi-channel pipetter, so they look like a little section of a 96-well plate. One side has the wells, which are the inlets for the assays, in other words, the reaction mixtures, and the other side has the wells that are the inlets for the sample. And what this chip then does is it makes all the combinatorial associations of samples and assays in these little tiny microchambers. There are, I think, 9 nanoliters each for, well, there's 2 chips. There's a 48 by 48 well, and there's a 96 by 96. And so you can see the scale of the reactions then for each of those is either 9 nanoliters or 6 nanoliters. So they're tiny, tiny volumes. And that's what the instrument does automatically for you, is to make all of those combinations. What that leads to then is 48 times 48, or 2,304 reactions for the 48 by 48 chip, 9,216 reactions for the 96 by 96 chip. And that's why I'm saying that if you had to individually pipette all of those combinations, it would be very, very error prone. Okay? But by only pipetting 96 and 96, or 48 and 48, It's order of magnitude less pipetting. Okay. So then what does the readout look like? Well, there's different levels of detail that you can get from the software that comes with this instrument. But at a sort of overview level, what you get is a heat map, essentially, where you have all of the different reactions corresponding to the different genes on one axis, and then you have all of the different samples, which in this case, for example, are single cells. So each sample is a cell. And then you can see, essentially, which genes are expressed. In this particular example here, there's a relative paucity of the genes that we're looking at that are actually expressed in these cells, although you can see there's a couple of genes, and these are the positive controls over here, which are always expressed. I should point out that the gap here is the no template control, so that's supposed to be blank. And then there are other genes that you can see are heterogeneously expressed. And again, that's the kind of information that you get out of this system doing single cell qPCR that you wouldn't get at the level of a bulk population of cell RNAs.
Does this actually go through the cycling to amplify the
Yeah, that's a good point. I should have mentioned. So when you have this chip, you first load it into one part of the instrument, which is basically making all of the combinations. So it opens and closes all these little microvalves and creates all of the reactions. And then you take it out of there and you put it into the cycler part of the instrument, where it then carries out all the qPCR and reads out the fluorescence. Yeah.
Is there a way to check from this output in the next slide if distinguish between a real negative and the reaction didn't work?
Right. That's a very good question. So for these genes, the question is there a way to distinguish a true negative from just the reaction didn't work? And so for all of these genes that have no positives anywhere, that's a very valid question, right? Did the reaction in fact work? And so in this example, we have no way to actually tell that. But what would be good is if you designed an experiment such that you had positive control samples for each of the genes you're reading out, or at least that you had validated those reactions with those particular probes in a previous experiment where they were positive. So that's what you have to do, and that's an excellent point. Because there are occasions where the chip fails, or perhaps we should maybe better say the investigator fails to properly load the chip. But in one way or another, if you get, for example, a little bubble at the bottom of your well, then it's not going to fill the microwells. And there's other kinds of things that could come up technically, and that could lead to essentially a negative result. So if that happens at the level of the sample, then you'll have something that looks like the no template control row, where you have just black all the way across. You know then that you can essentially discard that sample. But if it happens at the level of the gene, and if that gene is not expressed in any of the other cells, then it's hard to know whether it was the reaction or a technical error of the chip. Yes?
I have a question, and it might be a dumb question, but how do you load only like 6 nanometers?
Yeah, you don't do that. So what you do is you load an excess into the wells that look like the 96 well plate wells, right? And then the instrument has all these little micro valves that open and close and create in that little center portion all of the different nanoliter chambers. And they're filled basically just because they have a certain capacity and there's a suction applied, and so they fill to that capacity. Yeah. Uh-huh.
How do you know that you have a single cell and not two cells?
So, I was going to mention that a little later, but the technique is definitely dependent on good sorting, right? So when you sort single cells, you have to be essentially sure that you can deposit one cell per well. That's pretty routine now if you have a good cell sorter that has that microplate kind of capability. And if you set the right gates, for example, on singlets, with using your scatter parameters, you can be pretty assured that it's a single cell. More often than a doublet squeaking through, you might experience no cell, okay? Because either it didn't sort a cell at all, or the cell hit the side of the well somewhere and didn't actually get into the liquid and get the RNA extracted. We find here, we've worked with our FACS facility here, and they are really good about zeroing those cells right into the center of the well. And we get, from 48 cells, in this example you can see, they all showed something because all of the positive controls worked. And that's not too uncommon. If we do have 1 or 2 or 3 that were missed, that's about what that's about as much as we see. Yeah?
Have you tried C1, the new chip that they opened?
No, so I wasn't going to get into the company also has a device that can basically deposit single cells, although it's not a cell sorter like a fax machine because it doesn't interrogate those cells first to look at particular marker expression profiles. So we haven't used that instrument because we're always doing some kind of fluorescence sorting up front for particular phenotypes like tetramer positive cells or basophils or whatever, or B cell plasmablasts. But yes, if you had a homogeneous population of cells, or at least you didn't care to sort them on any phenotypic basis up front, then there is a device from the company that can deposit a single cell per well. Yeah. Go ahead.
You can sort directly into these plates?
Not into the plates directly. You sort into a conventional microtiter plate where you have essentially a buffer that lyses the cells and releases the RNA, and then you take that plate and do a preamp step, and I'll get to that in a minute, and then it goes into here. Yeah?
Normalization is required when using DEFT or data
I'm sorry, can you say? Normalization, yes. When you go beyond the step of just the raw data here, the next step is actually, the first next step is a QC to look at where reactions might fail, and I'll show you that in a moment. And then you probably want to do a normalization. So let me go actually to the next slide and address these points in a little more detail. So the workflow, which we were alluding to, is like this, at least for the concept where you're sorting the cells by FACS up front. So you first do your FACS staining and you sort single cells directly into this buffer, which is called Cells Direct, which is a lysis buffer that releases the RNA and protects it, so that you can then do a preamplification step. And in that preamplification step, you have the pooled primers of all of the different genes that you want to assess. And so it's a little bit, when I first was looking at these protocols, I thought, well, that's a little biased, right? But it's actually, it turns out that's a necessary step. If you don't do a preamp, you can't take one cell's worth of RNA and spread it into 48 chambers and expect to have a representation of every RNA in each of those chambers. You have to do a specified amount of preamplification first with the pooled assays. Then you've actually created enough material that you don't even have to use all of that preamped cDNA. You actually take a fraction of it, an aliquot, and load it onto the chip. Okay, so that's where you get to, from having just a single cell's worth of RNA that you could never recover by any other means to enough cDNA that you can load a chip and still have enough to go back and do 2 or 3 more times. Then you run your RT. This is in italics here because it's an optional step, which we actually don't do. And that's to run an RT-PCR first before you load the chip to see whether or not you actually have cDNA in each well. Okay? Because, as I said, sometimes your sorting might not be so good, and you might have a lot of empty wells, and so you could you could consolidate, and you could only put on the chip those where you know you have confirmed cDNA. But since we have a really pretty good success rate with getting cDNA, we actually find we can skip this step, and it doesn't cost us much. Then you prime the chip, so there's this device, as I mentioned, which essentially gets the chip ready first. Then you load the cDNA, and it makes all of the combinations. And you actually then, well, oh wait, I'm getting ahead of myself, sorry. You load the cDNA, you load the assays, the TACMAN assays, and then the machine makes all of the combinatorial, fills all the combinatorial chambers, and then you load that chip into the actual thermocycler where it does the qPCR reactions, and then you get your data.
Here's a question.
Yeah.
So when you're making the assay dilution plates, do you guys just make a big 96-hole plate master mix and then you go from that? Like, and where you can store it later on, and so you're not having to make up the dilutions every time? Because there's, valid
Yeah.
differences between assays if you're setting up one assay versus, your pipetting.
Yeah, it's not a bad idea, especially if you're going to do the same panel multiple times. I would think that that would be the way to do it. And
It's stable.
I-
The probe and with maybe the master mix.
I'd have to ask G about that, how long he can create such a mix and have it be stable. But certainly I thought maybe you were meaning just to array out on a plate the primer probe sets, but keeping it with the,
several plates or
yeah.
keeping it
I think for short term that would be absolutely fine. I don't know how long though. If you could, I wouldn't probably want to freeze and thaw that kind of a mixture multiple times.
How do you,
Go ahead.
How do you make sure that the cycling conditions are equal for each well?
Well, I mean, to some extent, they're all in very close proximity on this chip, right? So the instrument is built to cycle them all exactly together. I haven't seen that there's any kind of plate effects or edge effects that, within that small center of the chip where all the reactions are.
Are there any controls built in for that?
I mean, we've done things like made replicate reactions on two sides of the chip. And that essentially is a form of control for edge effects. And those have usually looked pretty good. So, okay, I've given you kind of a rough timeline here. I have to say this was actually figured out by Ophir Goldberger in the Davis lab a while back. So having not done it myself with my own hands, I can only say that's his estimate of the total number of kind of hands-on hours, which doesn't really count the data analysis because you can analyze this data many ways and for a long time. But it's essentially about a 2-day workflow. Okay. Oh, and there's the cost. Yeah, he says this part is priceless. There's the cost associated with it. If you talk about a 48 by 48 well chip versus a 96 by 96. And it seems pretty expensive for a single experiment, and I'll admit it is. You don't want to take on an experiment with 10 chips and just go at it without a lot of forethought because that's, $20,000 or $30,000. But I would also say that if you think about this as, it's not just a little tiny experiment we're talking about here. This is a few thousand qPCR readouts for each of these chips, right? So it comes out actually pretty close to $1 per qPCR reaction, which is not that much. All right, so now I want to show you a little bit of data, and yeah, I think it's okay that we run a few minutes over because we have a little bit of built-in breathing room in our schedule this morning. Other speakers, of course, will not go over. But I mean, I'm happy if it goes a little late because there's a lot of questions and a lot of interaction. That's the point. It's not that I want the speakers to drone on. So to show you a little bit of data, here is an example of two experiments that were replicates of each other with the same donor, the same input samples, actually. And we're just looking at how reproducible the platform really is. And I think if you look at this, you'd say, and I'll take it back. It's not the exact same input RNA. It's two experiments in which we sorted cells, but from the exact same donor. So row for row, the cells are different, but it's the exact same genes, the exact same protocol. And so the genes, for example, here that are showing up positive across the board are showing up positive across the board in both experiments. The ones that are always negative are always negative. There's a little bit of dirtiness here in this case, but for the most part, they're the same. And the ones that are heterogeneous in both cases, but because the cells are different, it's not going to be the exact same cell that lights up across two. So, but if you look at the general patterns, I think you would agree that it's relatively reproducible, with some few caveats. Like here, this is actually the slide I showed earlier. This no template control had a couple of positives. This one looks more clean, but yeah, this one. It's got a low-level positive in the no template control. Here's one cell that did not express this gene while all the others did. Probably that's just biological reality, but it's certainly possible that that's some kind of technical error. Usually if there's technical errors, though, as I mentioned before, you will see them across the entire plate, either an entire sample that didn't work or an entire gene reaction that either didn't work or shows positive for every single one. Okay.
You said that this is one individual subject,
Mm-hmm.
but two separate sorts,
Two different sorts, 2 different days experiments, yeah.
but one subject. So
One subject.
That column that's right before the positive control,
Yeah.
so that seems very different in the second experiment.
1, 2, 3, 4, 5, 6. There's like 6 positives here, and there's quite a few more here. That is true. Yeah, I hadn't caught that before. Is that just variability of sampling, or is it, something technical? We also haven't normalized. I mean, if you look, this is probably a lot brighter than this one on the positive control. So perhaps when we normalize, that signal might come down to about the same level. And that is something that we see. And it's actually good that you point that out, because here you can sort of tell that this whole chip is a little bit less intense, a little dimmer than this one. Where there's a positive here, it's brighter than it is over here. And if we normalize, we probably will take that difference away. Yeah.
When you're normalizing, do you follow always the same protocol? Like you can choose your threshold, you can do a global, for example.
We just do it to the positive control. Yeah, we do it per gene, and we do normalization to the control. Global normalization is a little dangerous, I think, because the cells can be different from one chip to the next. Yeah. Joanna, yeah.
So going back to the first step, when you do the sorting, so I can see how that's advantageous if you're looking at a certain subpopulation that you're trying to analyze. But if you're looking at a heterogeneous population, can you just do cell counting and put it in that buffer, or?
Sure, you could do, but I think you'd want to do a single cell deposition, like with this C1 instrument that they have, something that assures you have one cell. If you do just a limiting dilution, you're going to get, empty 2, 3 well, 3 cell wells. You'd want to be
So this is just to
precise.
accuracy of knowing that you have one.
Knowing that it was a single cell, yeah, that's really what it is. Yes.
So if you're normalizing to the bright positive controls,
Uh-huh.
wouldn't you be taking the chance of eliminating rare events?
DIM events, you mean, yes.
Rare gene expression?
Genes, yeah, that are expressed by,
Is there another normalization?
I mean,
Other than the super bright positive control?
just,
It works, but it's just too bright to really see.
I guess I would say that, you know, there's numbers behind all of this, and so for the most part, the brighter is better, the higher the number is better, the higher the expression, because what you don't want is to normalize to a value that's very near zero. Then you start to get a lot of heterogeneity that is just noise. But usually normalizing to a high value is not a problem. Although I guess your point is perhaps that at some level the positive control would saturate out. And then it's not a good normalization anymore.
Exactly right. You're essentially normalizing to a reagent control instead of an internal gene control, like a housekeeping gene.
Well, so those are housekeeping genes, but if they're expressed at such a high level that they're just, you know, a max kind of a readout, then it would be a problem. So I think, yeah, you want to choose your control and your amplification conditions so that your positive control is not completely maxed out.
This would, I think, only be a problem for rare events.
Yeah, I agree. Yeah, the dim signals on here.
Do you usually do a bunch of different housekeeping genes and then pick the average, or?
In this case, we've been usually doing two. We've been doing GAPDH and,
18S?
yeah, I think 18S. And I think the GAPDH is usually a little lower than the 18S. And so I think we like the GAPDH, but, well. All right. I'm going to show you in the last couple of minutes, and I'll try to do this quickly, just a little bit of data on a project where we were looking at these different CMV epitopes and the T cells that are specific for them across different donors and across epitopes from different CMV proteins. And we won't have to go into the details of the protocol, but basically we sorted single tetramer positive cells and ran the qPCRs. And I'm going to just show you a quick sampling of the data and the kinds of comparisons that we could do. So here, first of all, is the same epitope. This is a A2 restricted epitope of the PP65 protein from CMV. And I apologize, these are different slides from what's in your packet, but I updated them. And here we have essentially one donor with two different gene panels and a second donor with two different gene panels. And so just to orient you, these are kind of sideways from the other ones you saw. The genes now go across, and each column is a cell. All right, it's a complex picture to look at, but I think you can sort of scan over it and say, yeah, with maybe an exception here of a row that's different. Maybe you'll find another one if you look really, really hard. But for the most part, these patterns look very, very similar. They also point out a lot of heterogeneity cell to cell. There's only a few genes that are really, truly positive all the way across, more so in this panel than in this set of genes over here. But there's quite a few genes that are heterogeneously expressed across the cells, even though these are sorted tetramer positive cells. Yeah?
When you're analyzing these things, do you look at these visually, or does the machine sort of use an image analysis and just point out where there's, you know?
So we've been actually doing both. I mean, we'll look at them visually, but then you actually have, obviously, a CT value behind each of these. You have a whole CT curve, but you can, you know, establish a CT threshold and then quantitate it, and you can do statistics to say which ones are actually different. All right, so then if we instead ask the question, what about two different proteins that have T cells specific for different epitopes? So this one, I'm sorry, two different epitopes, but of the same CMV protein. So this is a donor. This is all one donor now. And the donor has T cells that we sorted that were specific for epitope 1, we're calling it, of PP65. And over here, we sorted tetramer positive cells that were specific for epitope 2. All right. Still basically the same kind of answer. They look really similar. Okay? And there's a lot of heterogeneity in both. So No big astounding differences so far. And there really aren't any hugely astounding differences in this whole story. But now I'm going to show you the last piece, which is what if we look at T cells, again, from the same donor, but now specific for an epitope of PP65, which is one kind of protein of CMV, and then an epitope of IE1, a completely different protein. So now we're looking at very different epitopes, not from the same protein. And again, it's still, the pattern is very similar, but I've now pointed out in the yellow boxes, about 6 or 7 or 8 genes that actually do have significant differences in the level of expression between these two epitopes. And so, again, you might ask, how do you actually quantitate this and decide what's different and what's not? Obviously, you can do an ANOVA. You can ask for significant p-values. But what we've also done is a PCA, or a principal components analysis, where each of the cells now is a dot, and these rings sort of encompass the boundaries of the cells within a given donor. And in this particular case, the blue dots are PP65 specific T cells and their gene expression. The red dots are IE1 specific T cells. And while there's a lot of overlap, there is some segregation where there's more of the PP65 specific T cells in this part of the PCA plot and more of the IE1s over on this side of the plot. But there is obviously some commingling, especially in some of these donors whose PP65 response looks really very much like their IE1 response. So that's kind of a way to summarize all of the data. And if you're not familiar with principal components analysis, it's basically a data reduction technique where we take all of the dimensions in terms of all of the gene expression levels for each cell, and we reduce them into 3 different components. In this case, it's a 3-dimensional PCA. And those components then are essentially conglomerates of the individual gene expression profiles, and they're the ones that best separate the 2 different groups. Or not the groups, but the samples themselves. And then as it turns out, they are somewhat grouped, where the IE1s are somewhat over here and the PP65s are somewhat over there. So it's looking for ways to separate the data in the fewest dimensions as possible. All right, so just to wrap up, I want to mention some things that we've kind of touched on. In terms of challenges, you need to have accurate cell sorting up front for this to work well. Or it's obviously not just for single cells. You can do bulk RNA, and it has all the other advantages of saving you pipetting and saving you reagents and allowing you to do large arrays of genes and samples. But you do need the right amount of input RNA. If you're way above or way below the targeted level, it's going to be hard to get comparative data. And you need to get the appropriate cycles of preamplification. It kind of goes hand in hand. If you use more input RNA, you need less preamp, and vice versa. And if you don't get that balance right, you're not going to have good readouts. In all cases, you have to get good at pipetting into the array so that you don't make bubbles and things. And you do have some challenges of how to look at all of this data. I'm going to go over that in just a second here, because you need to weed out the wells that had a poor reaction, and you need to pick a threshold value for your CT calculation. Here's one view that you can get of the data where you're looking at all of the CT curves superimposed on each other. And when you look at it this way, and this is a pretty good example of a plate run, you can see that the negatives really pretty much all stay down here, and by choosing a level here that's above this noise, you basically are only going to get positive CT values for things that actually amplified at some level. And you can see some of them, they're mostly amplifying in the range of about 17 to 33 cycles or so. And that's about the range that you want. If things start to come up after 5 or 9 cycles, they're way too abundant. If they don't come up until 35 or 38 cycles, they're you didn't have an F to get a good quantitation. All right, so that's basically the conclusions, which I've already mentioned, so I won't go over them again. The conclusions with regard to the tetramer study that I showed you are here, and basically just tell you that there's some genes that vary between epitopes, and even the cells specific for a given epitope can be quite heterogeneous, although they're similar across donors and across epitopes, at least within a single protein target. So these are some folks from our lab that worked on this tetramer project. It was led by Sheena and G in the lab. Sanchita has helped us with doing the normalizations and the analysis of the data. And the whole Fluidigm platform is in Mark Davis's lab, and Ophir and Evan were some of the early pioneers to help us get started with the technique. So I'll stop there. If there are any other quick questions, otherwise I don't want to get us too far behind. Go ahead.
Yeah, I would like to ask, do you normally do duplicates or triplicates, or you just run each sample once?
We usually do duplicates at least. Triplicates is great, but I think at least duplicates because of the possibility that you have, a blank column for.
Like when you have a very heterogeneous population, like how many cells you do? Like, because obviously 40 you cannot, you do 40, but if it's very heterogeneous, maybe you cannot even have a real.
Yeah, how many is enough? So the single cell, we haven't done the 96 by 96 on single cells. The company has sort of hedged a little on that. I think the well volume is so small that for single cell, you may not get a really reproducible answer. So we've done 48 by 48, and we usually do 48 cell, minus a couple of no template control and whatever. Maybe we'll do 2 chips worth for a single donor, but that's, it's partly a practical answer, right? How many thousands of dollars do you want to put into one donor? I think you can get good answers from 48 or 96 cells.
Maybe that's the last one.
Yeah.
Have you tried microRNAs apart from mRNA?
No, but that's something we want to do. We actually have that on our list. There's a project where we're going to try out some primer sets for miRNAs. Yeah. One last one.
In your epitope-specific gene expression profile in tetramer cell, across the donors, do you see any similarity? I don't know if you had done it.
Yeah. I actually showed the one slide of 2 donors, and we did, probably 8 or 9 different donors for that epitope, and very few differences between donors.